So, antibiotics. How do they work? Let's find out. So, if you watch my video on the history of antibiotics, you know that there was this idea about a magic bullet, right? Some chemical thing that could go into the body and kill the contagion without harming the host. And that there was lots of attempts, some of them more successful than others, at accomplishing this. The key concept in antibiotics is selective toxicity. Go here. Boom. Selective toxicity. Selective toxicity means that the antibiotic is more toxic to the bacteria or to the other pathogen than it is to the human host. Note that it's almost never going to be non-toxic to the human host. All of these things usually have some level of toxicity, but they work usually by targeting things about the bacteria or the fungus or the virus or the whatever, which are different from the human. The more different those things are, the better a drug this is. This is very different from the idea of, say, chemical control agents that are used to control in the environment. Things like iodine and bleach and alcohol and things like that, right? Those things might be useful at killing microbes, but they're usually just as good at killing humans as they are at killing microbes. Bleach obviously isn't something you would ever want to use on a human, but even if we get into things that are like antiseptics, such as alcohol, you might think, oh, well, lots of humans drink alcohol all the time. We use alcohol on humans all the time to get rid of microbes, and that's true in topical applications, on the skin, maybe in a wound. But those are areas where your cells are, generally speaking, already dead or damaged. It isn't getting systemic. If you had an infection that was spread throughout an organ, or that was within your blood, or even that was in, your subcutaneous tissues, killing it with alcohol would be very difficult. Trust me, you can't actually drink enough alcohol to raise your blood alcohol concentration to the point that would be necessary to kill microbes. I tried. You will kill yourself through alcohol poisoning before that happens. And alcohol is just as toxic to humans as it is to microbes, at least once it gets into your blood and into your body. So these organisms of selective toxicity, or these agents of selective toxicity, usually interfere with biological structures or processes which are common in bacteria, but not in humans. And while I'm going to use bacteria as an example here, and that is certainly the most common, the most common type of thing to use selective toxicity against. Selective toxicity has applications for any sort of human pathogen. And in my video on antibiotic specifics, I go into some of the targets of selective toxicity found in fungi and even viruses. So selective toxicity isn't a binary. It is a spectrum, right? Like, say, height or something like that, you can have more or less of it. So we have to have a way of measuring it. And the way that we measure the selective toxicity of a drug is through something called therapeutic index. And the therapeutic index of a drug usually is it's a measure of how selectively toxic it is. And it's the lowest dose that's toxic to a patient divided by a typical dose used for treatment. So it's a ratio of its toxicity to humans to its toxicity to bacteria or whatever. So, and just because something is more toxic to bacteria does not necessarily mean that it is that it's a better drug. Let me give you two examples here. Say we have a drug called pseudocillin, right? Which I'm going to do with a ES there. And with pseudocillin, the lowest dose that is known to be toxic to humans is 100 milligrams. And a typical dose that you would give to treat an infection is 50 milligrams. So here, the therapeutic index for pseudocillin would be 100 divided by 50, which equals 2. Now, say that we have another drug, and I'm going to call this falsamycin. And so with falsamycin, the lowest dose known to be toxic to humans is 1,000 milligrams. And a typical dose that you would take for a course of treatment is 100 milligrams. Objectively speaking, falsamycin is less toxic to bacteria, right? A typical patient would have to take twice as much falsamycin as pseudocillin in order to get the same result, not being sick anymore. However, also objectively, falsamycin is way less toxic to humans than pseudocillin is. So there's no problem giving a higher dose. Here, the therapeutic index would be 10. So which of these is a better, which of these is going to be a better antibiotic? This one. It has the higher therapeutic index, which means that the typical dose is a lot less close to the toxic dose. Generally speaking, you want to use the highest therapeutic index drug that you can successfully treat your patient with. So let's say that you've got a drug which is selectively toxic. It targets something about the bacteria that humans don't have. Say it's a pretty good therapeutic index, right? Not all antibiotics work the same way. You would think that, so some antibiotics can kill the bacteria that you're targeting, all right? We call those bactericidal. Bactericidal drugs kill bacteria. Cidal means to kill, like homicide or something like that, right? Some drugs are what we call bacteriostatic. These drugs do not kill bacteria, but they stop them from growing, usually from being able to reproduce. And then they depend on the immune system of the host to clean it up. You might think that bactericidal drugs would be preferable to bacteriostatic drugs. Like it's got to be better to kill the bacteria than to just halt it. But you're not necessarily right. Some of the best drugs that we have are bacteriostatic. Some of the best drugs that we have are bactericidal. In fact, it just depends. So, since bacteriostatic drugs don't kill bacteria, how could they possibly be better than bactericidal drugs? Well, think of a race, right? So all disease is a race between I don't want to say all disease, all infection is a race between your immune system and the pathogen. And if the pathogen wins, you die. And if you win, you kill off the pathogen. So at the beginning of this race, or sometime during this race, we're going to break the legs of the other runner. Who's going to win the race? You're going to win the race. Even I could win a race against somebody with 2 broken legs. And I'm not notoriously fast. So if you stop a bacteria from being able to reproduce, your immune system is gonna win. It's gonna take the thing out. Might not be right away, but, it's probably gonna be pretty quick. Unless you don't have an immune system. So bacteriostatic drugs, since they don't actually kill the bacteria and rely on the host's immune system, are not usually a great idea to use in a severely immunocompromised host. All right? Because even somebody with 2 broken legs is going to eventually pull themselves across the finish line if they don't have someone to race against. So so that's a circumstance where you might favor a bactericidal drug if you're dealing with a severely immunocompromised host. What might be a circumstance in which you would favor a bacteriostatic drug? Well, imagine somebody who had gram-negative septicemia, right? So they've got a gram-negative bacterial infection of the blood. What's going to happen if you treat them with a bactericidal antibiotic? Well, it's going to kill the bacteria, but it's a gram-negative bacteria. That means it's got endotoxins. If you give them a big old dose of bactericidal antibiotic and it goes in and kills all of the bacteria at the same time, you're going to end up with a huge load of endotoxin. And you could end up sending your patient into endotoxic shock. That might be a circumstance where it's better to give a bacteriostatic antibiotic. The bacteria is still going to die because the immune system's going to take care of it, but it'll probably happen over a somewhat longer period of time, so that instead of having a big spike of endotoxin, that happens when you give the antibiotic, you might have a lower, longer lasting load of endotoxin that happens as the immune system goes and cleans up the bacteria. Maybe this first one, right, all that much endotoxin at the same time is quite likely to produce shock. Over here, you might end up with a septic reaction, but something that is easier to deal with and easier to treat, right? That doesn't necessarily mean that all gram-negative infections are going to be treated with bacteriostatic antibiotics. There are circumstances where you might use a bactericidal one, but this is just an example of a circumstance where you might favor a bacteriostatic antibiotic. In addition to how they work, antimicrobial drugs also differ in what they work on. This is called their spectrum of activity. And generally speaking, antibiotics can either be broad spectrum or narrow spectrum. Generally speaking, broad spectrum drugs work on all or most bacteria. Specifically, they work on both gram-negatives and gram-positives. They might be static, they might be cidal, but they're going to work on all different types of bacteria. Narrow spectrum antibiotics will work on a subset of bacteria, but not all of them. Usually this breaks down to they'll either work on gram-negatives or on gram-positives, but not on both. There are also some narrow spectrum antibiotics that might specifically target acid-fast bacteria. So you might think that, you probably want to treat with a broad spectrum antibiotic. That's going to be better, right? And again, it kind of depends. You usually want to use the narrowest spectrum drug that you know to be effective. So if you're dealing with an unknown bacteria, like somebody comes in, they've got a septic infection, you don't know what's causing it, you're probably going to hit them with a broad spectrum antibiotic because you want to make sure that you kill it. Well, let's say the next day comes around and by this time you've determined that this is, Staph aureus. Staph aureus is a gram-positive organism. You would probably want to switch to a narrow spectrum drug against gram-positives at that point. Why would you want to do this? Well, if you recall back to our lectures on the immune system, the body's natural flora plays an important role in protection. So, a broad spectrum antibiotic is going to get rid of all of your body's natural flora. And that can sometimes have negative health impacts. For instance, if you treat somebody with a powerful bactericidal broad spectrum antibiotic, something like, say, ciprofloxacin, then you're going to kill off most of their gut flora. So most of the bacteria that live in your gut. And I say most because there's a type of bacteria that lives in most, not everyone, but most people have it, called Clostridium difficile, better known as C. diff. And C. diff is a endospore former. In an endospore state, bacteria are usually pretty much immune to antibiotics. So you're going to kill everything else. And then when you go off of the antibiotics, the C. diff is going to wake up. Say, hey, it's roomy in here. And you're going to end up with a massive bacterial infection of C. diff. C. diff is like something that's very common for people to have, but usually your gut bacteria keeps it in check. Get rid of the gut bacteria, and C. diff takes over, causing what's called antibiotic-associated colitis. And that's really kind of not a very descriptive term for what happens. The descriptive term would be explosive diarrhea. And it can kill people. And it can spread really easily from patient to patient. As if you become a nurse and progress through your career, you will almost certainly deal with C. diff at some point. And you will learn that words that I speak really cannot do it justice. And you don't want to deal with that if you don't have to. Another example of difficulties with broad-spectrum antibiotics, particularly in women, is a antibiotic treatment is frequently associated with yeast infections. Right? If you give a woman or other vagina-containing human a dose of broad-spectrum antibiotics, you're going to end up killing off the bacteria in the vagina. Those bacteria are usually acid fermenting, and they create a somewhat acidic environment in the vagina that is inhospitable to other microorganisms. Without them, the pH balance is disturbed. And worse than that, since it's an antibiotic and targets bacteria, it doesn't kill fungal cells. So the yeast will probably still be alive. And in the suddenly more hospitable environment has a tendency to take over, causing a really nasty yeast infection. So those are some reasons why, if you can avoid broad-spectrum antibiotics, it's sometimes better to do so. Now, when would you want to use broad-spectrum antibiotics? That's usually going to be whenever you don't know exactly what you're targeting and you need to treat quickly. Or if you're worried that there might be something in addition to what's obvious. Maybe the patient has been exposed to multiple potential pathogens. Or if you're delivering an antibiotic prophylactically, such as before surgery. You don't know what might infect them, so you'd probably better give an antibiotic that's good against everything. On that subject, so I just talked about a few of the adverse effects of antimicrobial drugs, suppression of normal flora being one of them, right? You kill off all of the normal bacteria, and that allows pathogens to take over. It reduces your ability to repel infections for a certain amount of time. There are a few other adverse effects that are possible. First, allergic reactions. This is particularly common with the penicillin class of drugs. Usually if you go to a hospital, one of the first questions they're going to ask you is if you're allergic to penicillin or if you have any drug allergies. And this is because penicillin is often, or I should actually say penicillin derivative drugs, are often the first prescribed, and they can be deadly if somebody has an allergic reaction to them, and something like a couple percent of the population does. Other things, aplastic anemia. This is particularly a feature of certain antibiotics. Some antibiotics have this effect, or can have this effect, but most don't. We don't know exactly what causes it. But there are a few antibiotics that, in a small percentage of the population, we're talking like 1 in 100,000, 1 in 1,000,000, right? Will just cause the red blood cells to all die, and then the patient dies. And there's really nothing to do about it. Like with aplastic anemia, usually the patient's gonna die. Unless they receive some sort of massive transfusion really fast. And there's unfortunately no way to predict ahead of time who's going to suffer from these effects. So drugs that have any possibility of causing aplastic anemia, even if it's very small, are often drugs that are just simply not used except in the most dire circumstances. Like if somebody's dying and this is the only drug you have that might work, then yeah, you go ahead and give it because they're dying anyways. Aplastic anemia isn't going to hurt them. But even though these drugs aren't likely dangerous, if I said, hey, I've got this drug, there's like a 1 in 100,000 shot, it's just going to kill you outright though. Would you take it? Well, probably if it was your only option. But what if I said, and we also have this other drug that has like a 0% chance of doing that. Which one do you want? Well, I want the one that isn't going to kill me. So that's the way that goes. Last adverse effect is antimicrobial resistance. This is something that is happening increasingly, particularly with some organisms. The first true antibiotic discovered was penicillin, and within 10 years of penicillin coming into wide use, there were varieties of staph bacteria that were resistant to it, and it became ineffective to treat them. So we developed other variations on penicillin. All of the other psyllin drugs, amoxicillin, ampicillin, methicillin, all of those things were often developed to get around this resistance. So like, we got to discover a new drug to kill off these bacteria that have suddenly become resistant. And usually within 5 or 10 years of an antibiotic becoming widely used, you start to have resistant strains develop. It doesn't mean that every strain is resistant, but some of them are. And the more we use the drugs, the more we encourage the growth of resistant strains. And this is true both on an individual, right? So the more we use a particular drug on you, the more likely it is that your bacteria will become resistant. I shouldn't say that. The more likely it is that resistant strains will become the dominant strain within you. But it's also true on a societal level. Like when we give you penicillin or methicillin or whatever, it doesn't stay in your body forever. And most of these are eliminated in the urine, meaning you just piss them out. And they're usually unchanged when they come out. And so they get into the environment where they help to breed resistant strains in the environment. And the more we prescribe antibiotics, the more antibiotics are out there, the more antibiotic-resistant strains develop. This was not a huge issue early on because we were discovering new antibiotics every few years. And as long as we kept getting new antibiotics, it wasn't a huge deal. But we've discovered like maybe one new antibiotic in the past 20 years. And only a very few in the 20 years before that. Like we've already plucked all of the low-hanging fruit from that tree. And Bacteria are continuing to become resistant, but we're not really discovering new antibiotics that fast. So this is a race that we're losing. The discovery of antibiotics is probably either the first or second most important discovery in human history, at least in terms of lives saved. Right? Antibiotics and vaccines together are the number one and number two discoveries in terms of lives saved. I couldn't tell you which one is number one, which one is number two. It's a little bit hard to say, but they're like up there neck and neck. And every year, because of antibiotic use, and particularly because of antibiotic overuse and abuse, one of those discoveries, antibiotics, becomes a little bit less useful. And eventually we're going to have a world where antibiotics are not necessarily dependable treatments anymore. And that's not going to be an awesome world. There is already a strain of tuberculosis which is immune to all known antibiotics. There is a completely resistant strain of tuberculosis, and it is present in the United States. Like, if you get this, we're basically moved back to, like, the type of medicine we had in, 1920. Because our antibiotics just aren't going to help treat it. We do actually have a few other non-antibiotic interventions that we can do, but they don't get rid of the bacteria. And so that's a thing to keep in mind before you just willy-nilly toss antibiotics out there into the wind. So as I said at the beginning of this lecture, all antibiotics, antimicrobials, depend on selective toxicity. And for bacteria, the normal mechanisms of selective toxicity all target things about the bacteria which are different from humans. These are the 6 most common targets. Cell wall synthesis. Drugs that target cell wall synthesis usually have very high selective toxicity, very high therapeutic indexes, and they are some of our best antibiotics out there. 2, protein synthesis. These target differences in the types of ribosomes that we have versus the types of ribosomes that bacteria have. Prokaryotes and eukaryotes have slightly different ribosomes. They're different sizes, they work through a slightly different mechanism, and they associate with different proteins. So that provides a whole plethora of potential targets to target. Some of these drugs are very high therapeutic index. Some of them are much lower therapeutic index because, if you think about it, are there any prokaryotic ribosomes in human cells? The answer is yes, because we have mitochondria, and mitochondria are descended from bacteria. And mitochondria have their own DNA, they make their own RNA, and they have their own ribosomes that can make their own proteins. So those mitochondrial ribosomes might be inhibited by these drugs. So nucleic acid synthesis. There are not very many drugs that target nucleic acid synthesis, simply, because there aren't all that many differences between how bacteria and we synthesize nucleic acids. So there aren't very many good targets there. There are a few. One targets DNA gyrase, which is an enzyme that bacteria use to help keep their DNA from coiling, whereas we use a different enzyme to do that works through a different mechanism. And another targets differences in the RNA polymerase between bacteria and us. But there aren't a huge number of great drugs in that category. Inhibition of metabolic pathways. So there aren't very many metabolic pathways that are both essential for life and different between bacteria and humans. But the most common one here is the metabolic pathway for folic acid synthesis. And bacteria have a completely different mechanism for synthesizing folic acid than we do. Actually, we don't really synthesize much folic acid. We mostly get it through our diet. But bacteria have to synthesize their own. So there's a whole class of drugs that inhibit this, and they usually tend to be fairly high therapeutic index drugs. Drugs that interfere with cell membrane integrity. There's unfortunately not a lot of difference between bacterial membranes and our membranes. They both basically work the same way. And so the drugs that inhibit cell membrane integrity are mostly very low therapeutic index drugs and are not safe for internal use in humans. Some of them are still fine for topical use. Like you can use them on a wound or on a skin rash or something like that because, they're not going to get into your blood. The cells on top of your skin are already dead. It doesn't really matter if you disturb their integrity, things like that. But these drugs can mostly not be used internally. And then the last category has to do specifically with tuberculosis and acid-fast bacteria. Acid-fast bacteria start off being resistant to most drugs, just by virtue of being acid-fast. A lot of these drugs don't work. Their cell walls are significantly different from both gram-positive and gram-negative cell walls. So most things that target cell wall synthesis don't work on them. A lot of the other drugs don't work on them either. So we have a special set of drugs that specifically work against acid-fast bacteria. And I go over all of these categories in more detail, referencing like the specific example drugs that I want you to know in my video on antibiotic specifics.